Good afternoon, students. We are going to continue with the types of fermentation. In the last class, we discussed about different types of fermentations like anaerobic fermentation, aerobic fermentation, then surface culture production or stationary fermentations, and submerged culture productions. So now we are going to continue with that. So we are going to discuss today batch fermentation, continuous fermentation, and dual fermentation. Now, what is a batch fermentation? As the name suggests, the products are obtained in batches in case of a batch fermentation. Okay, so it is a closed fermentation system. You have a sterile fermentation tank or vessel which we call as fermenter. So it's a closed fermentation system. The sterile fermentation tank is filled with previously sterilized mashed raw materials or substrate. Okay, so you have to sterilize the fermentation vessel. Then the fermentation vessel or fermenter is filled with sterilized raw materials, sterilized crushed, we call it mash of raw materials into the fermentation tank. Okay, all raw materials. Then the temperature, the pH required for the medium is properly adjusted, and intermittently, if certain additional nutrients are required, they can also be added into the tank. Okay, once you have added everything into the tank, you have to inoculate this raw material with appropriate inoculum. That is, you have to inoculate it with the microorganism that is going to bring about conversion of raw material into product. So you have to inoculate with appropriate amount of inoculum and allow the fermentation to proceed. Allow the process to proceed. Okay, after a proper period of time or after a definite time period required for the fermentation to convert substrate or raw material into product, you have to stop the product. So once the fermentation is complete, the end product that is formed is withdrawn aseptically from the fermentation tank, and the end product that you have recovered from the fermentation tank is further processed for recovery, for product recovery, then packaging, marketing, etc. Okay, so you have a tank which is a sterile. It is filled with sterile nutrient medium, that is raw material. It pH, temperature, everything, nutrients, etc. are all controlled, and then we allow the inoculate it with the organism and allow the fermentation to proceed. Once the fermentation is over, take out the fermentation product that is filled with the tank. You take out the fermentation product, then proceed for recovery, packaging, etc. The fermentation, if once the fermentation is complete, the whole vessel is cleaned and sterilized. Okay, the vessel is cleaned, sterilized, and the whole process is again repeated. So this is continued. One fermentation run, after one fermentation run, collect the product, then wash the vessel, sterilize the vessel, again proceed. So again and again this process is repeated. Thus each fermentation is a discontinuous process and is divided into number of batches, and hence the name batch fermentation. Okay, in case of batch fermentation, the composition of the medium is not constant throughout the process. In the beginning, the concentration of raw material or substrate is more, and towards the end of the fermentation process, the concentration of product is more. That means the composition of medium is not constant throughout. Because of this, organisms exist in all growth phases of their life cycle. That means lag phase, logarithmic growth phase, stationary phase, decline phase, death phase, and all. Okay, so you cannot maintain, because the composition of medium is not same, you cannot maintain the organism in logarithmic growth phase. That means the physiology of the organism is constantly changing with time. Due to this change in physiology of the organism, steady state conditions cannot be achieved. Okay, in batch fermentation we cannot achieve steady state condition, and hence regulation and automation of this kind of process is not possible. So the process cannot be automated. Now, if you compare batch fermentation with continuous fermentation, the rate at which the product is produced, that is production rate, is less as compared to continuous fermentation in some cases. And there are chances that the organism may undergo mutation, and hence the fermentation may get contaminated because of the organism undergoing mutation. Okay, so this is about batch fermentation. You can look at the fermentation tank here. It's a tank. We call it bioreactor or fermenter or like that. So there is a agitation device. This is the motor of the agitation device. These are the impeller blades. Okay, agitator which moves. You can add the medium. You can control aeration rate, pH, temperature, etc. Allow the fermentation to proceed and collect the point where it is written effluent. It is the discharge point from where you can collect the fermented broth or fermented medium. Okay, so this if you look at the graph here, what happens with time as the time proceeds? Okay, if you look at the graph, you can see that as the time proceeds, the concentration of reactants go on decreasing. Okay, the concentration of reactants go on decreasing as the time proceeds. That means concentration is not same throughout the process, right? Now let us discuss about continuous fermentation. In this type of fermentation, the end product is recovered continuously, and hence the name continuous fermentation. End product is recovered continuously. In this method, fresh nutrient medium is added continuously or intermittently into the fermenter, and equivalent amount of fermentation end product is withdrawn from other point continuously or intermittently. Why do we call it a continuous fermentation? Because from one point, or you can say from the addition point, the fresh nutrient mediums are continuously or intermittently added, and from the exit point or from the recovery channel, we go on collecting the fermented end product, okay, continuously or intermittently. That is why the name continuous fermentation. So naturally the rate at which the product is produced is also most of the time faster. Okay, and it's not a discontinuous process, it's a continuous process. We go on continuously adding the nutrient from the addition point, and we go on continuously collecting the product from other end. The most important precaution that we need to take here is that the rate of inflow of raw material must be equal to the rate of outflow of products. Okay, the rate at which nutrients are added inside should be similar to the rate at which the products are collected from the exit point. Okay, so rate of inflow and rate of outflow is to be controlled. So you can see here, this is a fermentation tank. Okay, and this is a nutrient medium reservoir. With the help of the pump, the nutrients are continuously added, and after a particular time, the products are continuously recovered. This is actually an overflow tank, and but the products are continuously recovered. Okay, so this is nutrient reservoir. We go on adding the nutrients to the main fermentation vessel. Okay, and from the other point we go on collecting the product. Now look at this. So fresh medium from reservoir is added, sterile air, flow regulation and all in all, everything, temperature, pH, everything is properly controlled. And from the outflow point, we go on collecting the product. Okay, right. That is about continuous fermentation. Now here, steady state conditions can be achieved. The steady state conditions can be achieved in continuous fermentation either by using a chemostat or by using a turbidostat. That means either by using chemostatic technique or turbidostatic technique, we can maintain steady state conditions. That means the physiology of the organism will remain same. Organisms will constantly remain in logarithmic growth phase, exponential growth phase. Why organisms can remain in logarithmic growth phase? Because the concentration of the medium is constant. Okay, there is no change in concentration of the medium that is present in the fermentation vessel, because there is continuous addition of nutrients and continuous removal of products. So the concentration of the medium as a whole is constant, and hence we can achieve steady state and maintain the organism in logarithmic growth phase. Because of this, regulation as well as automation of the process is possible. Okay, continuous fermentations save production time and they also require less space. So they are, in one way, they are superior to batch fermentation because they can be run continuously and we can go on collecting the product. So time required as well as space required is less. Now, a continuous fermentation is generally carried out in three ways. Number one, single stage fermentation. Number two, recycle continuous fermentation. Number three, multiple stage continuous fermentation. So, let us discuss one by one. Single stage fermentation. In single stage fermentation, there is a single fermentation vessel inoculated with the inoculated with the organism, and the nutrient medium and culture are kept in continuous operation. Okay, you go on adding the nutrient, you go on adding the culture of the microorganism, and you continuously maintain the operation by balancing the input and output. That means inflow of raw materials and outflow of fermented end product. Okay, so from one point nutrient mediums are added continuously and from another point the fermented broth is harvested. So the balance is kept and hence we can carry out the fermentation continuously. But the vessel here used is a single vessel. That is why the name single stage fermentation. Now recycle continuous fermentation. See, there are certain fermentation medium or fermentation raw materials which actually are deficient in carbohydrates, which are deficient in carbon compounds, and hence microorganisms tend to grow very slowly in such raw materials. So what we can do is we can add certain microorganisms from the previous cycle into the fermentation vessel. Right. So microorganisms from the previous batch, from the fermented previous batch are added into the new batch of raw materials. So that is why the name recycle continuous fermentation. In this way, in this method, a portion of the medium, fermented medium, withdrawn from the vessel is added back to the culture vessel. Thus, the culture is recycled to the fermentation vessel. This method is generally adopted in hydrocarbon fermentation processes. Because of recycling, you are actually giving a lot of microorganisms to the, you are adding a lot of microorganisms into the fermentation medium. The recycling will provide cells at a higher population in the fermenter, which results in great or enhanced productivity of the desired product. Okay, so in recycle continuous fermentation, a portion of fermented medium is recycled back to the main fermentation vessel. It is adopted generally in case of nutrient media like sulfite waste liquor which are deficient in carbohydrates. Okay, recycling of cells provide higher microbial population so that it results in enhanced productivity of the desired product. So that is recycle. We recycle a bit of fermented broth into the vessel, back into the vessel. Number 3 is multiple stage continuous fermentation. In this process, 2 or more fermentation vessels are employed. Okay, in single stage there is only one single fermentation vessel, that is one tank, one fermenter. In recycle also there is a single fermenter. But in case of multiple stage continuous fermentation, 2 or more fermenters, 2 or more fermentation tanks are employed simultaneously and the fermentation is operated in a sequence. Why do we need 2 or more fermentation vessels? Different phases of fermentation processes like growth phase and synthetic or production phase are carried out in different fermenters. Okay, in multiple stage. Say for example, if you have 2 tanks, then growth phase of organism where the organism only grows and multiplies. That means the population of microorganisms go on increasing. That is growth phase. So growth phase is maintained in one vessel, and synthetic phase. Synthetic phase is the phase in which The high population or high number of microorganisms together, they convert the substrate into product. That is, synthetic phase is a production phase where substrate is converted into product. So multiple stage continuous fermentations are those fermentations in which more than one fermenter, more than one tank is used. Okay, different phases of organism like growth phase and synthetic phase are carried out in different fermentation vessel. Generally, growth phase is allowed in first fermenter and synthetic or production phase is allowed in second and subsequent fermenters. If you are third also, then synthetic phase in second and third fermenter. This process is adopted particularly in those fermentations wherein the growth phase and synthetic activities of the microorganisms do not occur simultaneously. Okay, if there are most of the fermentation in which the organism grows, multiplies, and while it is growing and multiplying, it is converting the raw material into product. But there are certain fermentations in which the growth activity and production activity is different. Okay, it does not occur simultaneously. So in those cases where the growth and synthetic activity of the microorganism are not simultaneous, okay, synthesis is not growth related but occurs when the cell multiplication has stopped. In such cases, we use multiple stage continuous fermentation. Okay, why? Because growth and synthetic activities do not occur simultaneously. Production or conversion of raw material into product occurs only when the growth and multiplication of the cells have stopped. That means growth activities and production activities are different. So in that case, we use multiple stage continuous fermentation. That means growth is carried out in one vessel and synthesis is carried out in another vessel. And as I told you earlier also, in case of continuous fermentation, we have to maintain steady state. Steady state means organisms will remain, all organisms inoculated into the fermentation medium will remain in logarithmic growth phase, exponential growth phase. And how can we maintain them in the exponential growth phase? Either by turbidostatic method or by chemostatic method. Okay, so that is all about continuous fermentation. Now let us discuss about dual or multiple fermentation. Okay, dual or multiple fermentation. What are those? Dual or multiple fermentations are those in which more than one microorganism is used. That means we are not going to use a single microorganism to convert the raw material into product. We are going to use here more than one microorganism. Okay, say if you are using two microorganisms, right, then the organism used may be inoculated simultaneously into the growth medium. That means at the same time you will inoculate organism number 1 also and organism number 2 also, or you inoculate the medium with organism 1, allow it to grow, and then inoculate or add the second and allow it to grow. Okay, so the two organisms may be inoculated simultaneously into the growth medium, or one organism is grown first in the medium and then inoculate and grow the second organism into the medium. Alternatively, what you can do is grow one organism in a separate vessel, grow second organism in a separate vessel in the nutrient medium, and after the two have grown separately, then you have to mix the two fermentation or combine the two fermentation for further fermentation activity. Okay, so in one vessel, one organism number 1 will be grown and allowed to carry out fermentation activity. In vessel number 2, inoculate the second organism, allow the fermentation activity, then combine the two fermentations to get the or for further fermentation activity. This is how dual or multiple fermentations are carried out by using two or more microorganisms. Why do we need two or more microorganisms for any process? The basic concept of a dual or multiple fermentation is that two or more microorganisms can accomplish a change which neither organism can do alone. That means if organism number 1 is grown, it will not give you the desired product. If you inoculate only organism number 2, it will not give you the desired product. But together, organism number 1 and 2 together can give you the required product. That means the two organisms can accomplish a change, can bring about a change which neither of the organism can bring alone. No, none of the two organisms can do the production alone. Okay, so they require help of one another. That is why We need two organisms, two or more organisms. The most obvious use of this kind of fermentation is use one first organism to produce a fermentation product, which is then changed by the second or further microorganism into a different fermentation product having greater economic value. Okay, so organism number 1 will produce a fermentation product. This will serve as substrate for organism number 2 or 3, whatever. So organism 1 produces fermentation product. This fermentation product is then converted by second or subsequent organism into a third different product, and the last product that is formed has a greater economic value. Say for example, vinegar production, acetic acid production. Okay, what happens in vinegar production? Yeast, that is Saccharomyces cerevisiae, produces ethanol anaerobically from molasses. Okay, ethanol is produced, or rather you can say that yeast converts molasses into ethanol anaerobically, and then acetobacter oxidizes ethanol aerobically to give vinegar, right? This is how it's a dual fermentation. That means acetobacter directly cannot convert molasses into vinegar. Acetobacter is required for synthesis of vinegar, but acetobacter cannot directly convert molasses into vinegar. So what do we do here? First, yeast, that is Saccharomyces cerevisiae, is grown on molasses, right? Under anaerobic condition, molasses will be converted into ethanol by Saccharomyces yeast. Okay, and then this ethyl alcohol is oxidized by acetobacter into vinegar, that is acetic acid. The combining of separate fermentation to yield further fermentation activity has wide industrial application. Okay, so in this fermentation approach, one fermentation yields a product that must be enzymatically converted into second product of greater economic value. Okay, in this time, one fermentation yields a product that is enzymatically converted into second product of greater economic value. Let us take another example of L-lysine production by dual fermentation. Okay, now E. coli auxotroph is written here in this reaction. Auxotrophs are nutritionally deficient organisms. Okay, they are deficient in one or the other nutrition. So, E. coli auxotroph cannot convert directly glycerol into L-lysine. So, what will E. coli auxotroph do? E. coli auxotroph will convert glycerol into alpha epsilon diaminopimelic acid. Okay. So, E. coli auxotrophic mutant converts glycerol into alpha epsilon diaminopimelic acid. Right. Now, this organism E. coli auxotroph does not have decarboxylase enzyme because diaminopimelic acid is decarboxylated to remove CO2 so as to get lysine. But the organism is an auxotroph. It does not have decarboxylase activity, and hence we require a second organism, that is Aerobacter aerogens. Okay. This organism produces an enzyme DPA decarboxylase, that means diaminopimelic acid decarboxylase, and brings about decarboxylation of diaminopimelic acid and synthesis of L-lysine. Okay. So glycerol is converted into alpha epsilon diaminopimelic acid by E. coli auxotroph, which is then converted into L-lysine by the activity of enzyme DPA decarboxylase by Aerobacter aerogens. So it's a dual fermentation because two organisms are utilized. Auxotrophic mutant of E. coli is used in the first half of the fermentation, and wild type or prototrophic E. coli or Aerobacter aerogens is employed in the second half. So in the first reaction we use E. coli auxotroph. In the second reaction we utilize Aerobacter aerogens. Okay. Diaminopimelic acid produced in the first half by the auxotrophic mutant of E. coli is converted into L-lysine by Aerobacter aerogens in the second half. Okay. So that is the basic check. Now another approach is that of dual fermentation is that one organism produces a change in the medium such that it becomes suitable for the growth of the second organism. One organism provides growth factor for the growth of another organism. One organism removes toxic materials so that second organism can grow. One organism removes oxygen so that and reduces the redox potential so that the second organism, which is anaerobic, can grow in the medium. Is that clear? One organism maintains pH in a critical range required for the growth of the second organism. One organism produces a metabolic product like lactic acid, which is both beneficial for the growth of the second organism and checks contamination also. So employing two or more microorganisms has some significance, that one organism may be creating proper growth conditions for the second. One organism is removing oxygen for the second, removing toxic material so that the second can grow. So like that. So dual fermentations do have significance. Okay. Now how do we grow the organism? In the beginning, I told you that the organism can be inoculated simultaneously into the medium. But simultaneous growth of two organisms in a single medium is very difficult to control. Okay. And extensive research and studies are required to balance the growth of two organisms simultaneously. Okay. So simultaneous growth of two organisms is very difficult to control and we require extensive research. So most of the time, dual fermentations employ growth of one organism followed by inoculation and growth of the second organism, and it is easy to control it. Okay, if we inoculate the two simultaneously, it will become difficult to control the growth of both at optimum. So better, easier way of controlling the process is inoculate the first, allow it to grow, and then inoculate the second, and growth activities of the second are followed by the growth activities of the first. Okay, and this is particularly true if it is possible for us to kill the first organism by heat or any other method and inoculate the second organism. Okay, so dual or multiple fermentations are those in which two or more microorganisms are used to accomplish the synthesis of a desired product, right? So that is all about types of fermentation. Next time we will discuss about design and configuration of a fermentation vessel.